So, first of all, I would like to congratulate Dr. Yannick Sava on the kickoff of this new institute, and I would like to thank him not only for the invitation to talk here, but also for the invitation to join the institute as a new faculty, as a founding faculty. I'm very honored by this, and I'm very excited and very motivated, and which somehow brings me to the topic of my talk. So, all of you probably like to drink coffee and also tea and sometimes you may wonder why coffee actually wakes you up, and I hope I can give you an answer in the next 15 minutes on that. The sleeping habits of humans are quite unique in a sense that we can defy sleep and stay awake for recreational and occupational reasons, and the motivation to stay awake is often supported by the use of psychoactive drugs that includes prescription medicines, that can include also, of course, drugs of abuse. But most prominently is here the use of this compound here. This is caffeine, which is of course in coffee, as already said, in tea and in many other beverages, also in cold medicine. So this makes the world's most widely used psychoactive drug, and it's causing wakefulness by blocking the action of adenosine. And when we talk about adenosine and sleep, then this is a very enduring story that actually goes back to the 50s, when a group in London, they injected adenosine into the CSF of cats, and they found that when they do that those animals, they go into a state that resembles sleep. And then over the years, it was discovered that adenosine can act on two types, basically, of adenosine receptors, A1 receptors and A2A receptors. One of the landmark discoveries was in 1997 by Porter Heiskanen in Bob McCully's group at Harvard Medical School. They found that adenosine accumulates during a prolonged period of wakefulness in the basal forebrain, mostly. And then they later showed that this adenosine that accumulates in the basal forebrain is acting on adenosine A1 receptors, which are inhibitory and thereby blocking the release of acetylcholine into the cortex and causing sleepiness. And our group also has recently shown that those adenosine A1 receptors also expressed on histaminergic neurons in the TMN, and the same effect, if they block the release of histamine, they cause sleepiness and inhibition of wakefulness. Our group is also very interested in those adenosine A2A receptors because there's a simple fact: if you induce or infuse an agonist for this receptor into the CSF, the animals will fall asleep. And we are mostly interested in adenosine A2A receptors because it provides a link between the human regulation caused by prostaglandin D2 and the neural system of the brain. And for many years, the $1 million question was, because caffeine is an antagonist for both receptors, for the A1 receptor and for the A2A receptor. Really, the $1 million question was, which of the receptors is responsible for the arousal effect of caffeine? Is it the A1 receptor or the A2A receptor, or are both receptors required? So a colleague of mine at the Osaka Bioscience Institute, he used knockout mice for both receptors, and he gave them caffeine, which is very unusual. So in this case, the animals, if you do this in wild-type mouse, the animals, they wake up and they stay completely awake for almost 3 hours. There's no sleep at all during that time. And this is, of course, done during the daylight when the animals are mostly asleep. And if I said already, do this in a wild-type mouse, they wake up. But the same happens if you do this in a A1 receptor knockout mouse. It clearly shows that the A1 receptor is not required for the arousal effect of caffeine. However, if you give caffeine to a knockout mouse for the A2A receptor, you will see there is no effect to caffeine whatsoever. It shows that the arousal effect of caffeine depends on adenosine A2A receptors. Now, the limitation of these experiments is that you cannot further localize the brain area which is required to mediate this arousal effect of caffeine through adenosine A2A receptors. And that's when I came into that project, and we want to know which area is responsible for that, and If we talk about adenosine A2A receptors, the first interesting area is of course the basal ganglia, because that's where the adenosine A2A receptors are most densely expressed. So this here is immunostaining for A2A receptors, and you see here the brown is a dense expression of A2A receptors in the dorsal part of the striatum, the cauda putamen, but also in the ventral part, including the nucleus accumbens, the shell and the core. Now when it comes to research on the basal ganglia, this is a very crowded field. And over the years, this area has associated with many important physiological functions. Motor control in the dorsal striatum, procedural learning, habit formation. Nucleus accumbens is involved in reinforcement, reward, motivation, locomotion, initiation of locomotion. And also the most ventral part here is also still important for reward, initiation of locomotion, but also sensory perception. However, the role of this area in sleep-wake regulation is unclear, and there's almost nothing known about that. And to address the question if adenosine A2A receptors in the basal ganglia, especially in the striatum, are responsible for the arousal effect of caffeine and may have a role in sleep-wake regulation, we used two approaches to knock down the A2A receptors. One is a mouse model in which we used the floxed mouse for the A2A receptors and used a viral vector that expresses greater combinase to focally inject this virus into brain areas, in particular the striatum, to get a knockout deletion of these A2A receptors. And the second approach that we used, that was done in rats, in this case we used a system based on RNA interference, and again we used the viral vector that expresses short hairpin RNA that gets within the neurons converted into interfering RNA, leads to the cleavage of the messenger RNA, and thereby to loss of function. So this viral vector also contains a reporter gene and Cherry to visualize the location of the viral infection. And what I just want to show, this is our, of course we are interested to measure wakefulness, but also sleep. Then we use a sleep bioassay system. It's just briefly shown. I think I can skip mostly since we are all sleep experts. So I just want to say that what we have in our institute at Osaka Bioscience is a cable-based system. So we put screws into the skull and electrodes or wire into the muscle to measure EMG, and for the screws to measure the EEG. We also have locomotor activity, and thereby we can distinguish between the stages of wake, non-REM, and REM sleep based on the EEG, EMG, and locomotor activity. And because my talk is very short, I don't want to go into details how to do that. I assume that everybody knows that. So this is basically, these are the data shown based on the viral injection of a Cray-expressing AAV into our floxed mouse. So what we see here is if we inject, if we knock out the A2A receptors in the nucleus accumbens. So what you see here, this is a mouse, a floxed mouse injected with AAV Cray, and the control mouse is also a floxed mouse that is injected with a virus that expresses mCherry. So in the control mouse you have a normal expression here of A2A receptors in the nucleus accumbens, whereas in those what we call the nucleus accumbens knockout mouse, we have a deletion of the A2A receptors here in the nucleus accumbens. And if we look at the arousal effect of caffeine in those mice, and we see that in those control mice, they have a normal response to caffeine. So if we give caffeine here at a dose of 15 milligram per kilogram, we see a strong increase in wakefulness that lasts more than 2 hours. And however, if we give caffeine to a mouse that does not have any A2A receptors in the nucleus accumbens, we see that the arousal effect of caffeine is essentially gone. And we also we repeat those experiments, as already said, by using a rat model in which we used RNA interference to knock down the adenosine A2A receptors. And this is just the essence of these experiments. What you see here is a knockdown of A2A receptors in the nucleus accumbens, core versus shell. So here in this rat, we have permanently knocked down the A2A receptors only in the shell portion of the nucleus accumbens, whereas there's almost normal expression of A2A receptors in the core portion. On the other hand, in this rat, there is a knockdown of the A2A receptors here in the core portion, but the normal expression of A2A receptors in the shell portion. And if we look at the arousal effect of caffeine, we see that if we give caffeine to those rats, we see that if we knock down the A2A receptors in the core, there's no attenuation of the caffeine effect, the normal effect of caffeine, 3 hours here of wakefulness, whereas we knock down the A2A receptors here in the shell portion of the nucleus accumbens, we see an attenuation of the arousal effect of caffeine. So that clearly shows us, and then we concluded that adenosine A receptors here in the nucleus accumbens, and especially in the shell portion of the nucleus accumbens, are responsible for the arousal effect of caffeine. Now, if caffeine is effective, for caffeine to be effective as an antagonist, that means that those adenosine A2A receptors must tonically be activated by adenosine, otherwise there's no effect. And that brings us to the question if excitatory adenosine A2A receptors in the nucleus accumbens restrain arousal system and cause sleepiness. And to answer that question, we used 2 in vivo approaches for conditional activation of neurons. And the first approach is based on a TREAD system. TREAD stands for designer receptors exclusively activated by the designer drug. What we have here, these designer receptors, these are human muscarine receptors which are mutated, so they cannot be activated anymore by the endogenous ligand, which is acetylcholine, but they can be activated by an artificial substance, which is this dopamine agonist metabolite, clozapine N-oxide, which activates the receptors, and there are 2 types, HM3, which is a GQ couple, so an excitatory receptor, and they are also HM4, which are GI coupled, so they would be inhibitory. We have mostly focused on the excitation of the system. And this TREAD system is in a flex system, so this is conditional. This is a viral vector here. That means in the basic, the system is inverted, so the virus cannot express an active system. However, in the presence of critical combinase, the whole system gets inverted, and we get an active expression of the TREAD system. And what we do is we inject those viral vectors in a transgenic mouse in which the critical combinase is expressed by the adenosine, is expressed under the control of the adenosine A2A receptors. That means it's only in the striatum, it's only activated in the neurons of the indirect pathway. And running out of time, so I have to go quickly. This is the normal EEG during the night when the mouse was injected with saline, and we see here normal EEG. So this animal is awake, there's locomotor activity, muscle activity, and you see here the animals are mostly awake. However, if we inject in those mouse, which has this TREAD system in the striatal neurons of the nucleus accumbens, and we inject CNO, then we see a strong induction here of slow wave activity. Animals, they go asleep, and they sleep during a period of almost 2 hours. And this is just the quantitative summary. If what we see here is that, first of all, here this is the expression of the TREAD system in the nucleus accumbens. And if we give CNO versus vehicle to those mice, we see a strong induction of non-REM sleep when those neurons here are activated by the TREAD system. And in the second approach, we also use the optogenetic method to activate those neurons. In this case, the viral vector expresses channel rhodopsin, which can be activated by blue light. And again, it's a conditional viral vector that we injected into this transgenic mice that expresses critical combinase under the adenosine A2A promoter. And we stimulate with this protocol basically in a period of 2 seconds. We stimulate in the first quarter at a frequency of 40 Hertz with a duration of 10 milliseconds. And as a control, we use blue light because this channel rhodopsin cannot be activated by we produce yellow light, sorry. And again, here, this is what we see in the normal mouse. In the mouse geronopsin nucleus accumbens, we see the yellow light, normal sleeping, normal wake animals with no sleep at night. However, if we use blue light to stimulate nucleus accumbens neurons during the night, we see induction of slow wave activity and the animals that go to sleep, which is very unusual for an animal during that period of time. And this is just a summary. It shows that that when we stimulate with blue light, we see induction of non-REM sleep. If we stimulate with yellow light, there is no induction of REM sleep. The animals continue to stay awake. And this is just the conclusion. What we think so far, that the nucleus accumbens is a key structural element for the control of sleep and wakefulness. And we think that brings me back to my beginning, that the motivation state is an important fundamental regulator of sleep and wake. And here, thank you very much.
Thank you. We have time for more questions. So in the meantime, Shara.
Very much interesting. Have you tried to characterize through using the power spectrum, if the sleep that you induce through the activation, the optogenetic activation, does it look like normal sleep in terms of, for instance, low waves?
It's essentially normal sleep. So the power spectrum is identical to normal sleep. There's no significant shift or whatsoever in the power spectrum.
Robbie Green.
Yes, so I'm just wondering, so the one question might be then whether
Use the microphone.
Okay, thank you. One question might be whether these neurons then are important in actually mediating the transition from waking to sleep. In other words, do they have to become active in order to make that transition, or do they normally become active? I was wondering if there's any CFOS evidence for such a thing happening.
It's difficult to see any CFOS during normal sleep-wake cycles. Only what you see is CFOS activation if you give them an agonist for the A2A receptor that induces strong CFOS expression in the nucleus accumbens, besides other areas. But of course, this is an artificial activation of the receptors.
Exactly.
But during the normal cycle, there is no clear CFOS expression in the nucleus accumbens. So
do you have any sense, or do you have any thoughts about what kind of circumstances might result in activating these neurons and facilitating this transition?
So what I can say is, if we knock down A2A receptors in the nucleus accumbens, what we see is in the baseline during the night, the day animals are very more awake in the initial hours. So that's basically when the animals basically just wake up, become active, start probably looking for food, and then something like this. So, and then the knockout mice are more awake than normal mice. So that means that somehow this motivation-driven or motivation, or when dopamine levels increases during a period of active locomotion, that this also leads to maybe kind of dampened by the adenosine system. So I'm not sure if this adenosine system is more important during the day or during the night. So during the night might be just basically to counteract the dopamine system.
Like a brain.
Yeah.
Okay, thank you very much.